Welcome back, everyone. In the previous segment, we talked about the major sequence and structure databases and how to search them. But of course, there are hundreds of additional biological databases for various purposes in basic science, agriculture, biotechnology, and medicine. Today, I will describe several databases that focus on variation within the human genome. Such databases have many clinical applications, such as identifying pathogenic mutations, calculating risk score for complex diseases, determining drug dosage, and tailoring treatment for cancer. I will then introduce genomic browsers, graphical interface that allows users to browse through the human genome and explore the many layers of information that can be presented in the browser, including information about human sequence variations. Humans are diverse. No two people are identical. Actually, even monozygotic twins have several dozen differences between their genomes. The Human Genome Project was based on sequencing the genome of a small number of individuals to create the reference human genome. This reference genome is like a consensus, but each one of us differs from the human reference genome by about 1 nucleotide per 1300, which amounts to millions of changes throughout the genome. The coding regions of the genome are more conserved, but still we have thousands of nucleotides variation even in our coding genes. It is clear that our unique individual traits are a result of these variations. Most of these variations are changes in a single nucleotide, although some affect larger segments of the genome, as in duplications or deletions. Mutations are introduced randomly into the genome during reproduction. As we will discuss in later units, because of evolutionary selection, some mutations become fixed in the population, while others are eliminated. dbSNP was developed as a repository of mutations that are relatively common, present in more than approximately 1% of the human population. The assumption was that if a mutation is relatively common, it is likely not deleterious, as otherwise it would have been eliminated. Thus, these changes were called SNPs, single nucleotide polymorphism, leading to innocent human diversity. Later, it was realized that there are mutations that are quite common, but still may have negative effects on health, for example, in conjunction with other confounding mutations. Thus, we now prefer to use a neutral term SNV, single nucleotide variant, though the name of the database was not changed, and it is still dbSNP. Currently, dbSNP contains information regarding more than 100 million validated SNVs and many more that are not considered validated. These SNPs are mainly reported by large sequencing projects, regardless of whether or not they are pathogenic. For each entry, dbSNP reports its genomic location, information about the nature of the mutation, whether the variant is expected to lead to a change in the protein amino acid sequence or a splicing change, and estimation of the frequency of this variation in human populations, according to several large-scale human genome sequencing projects like the 1,000 Genomes. As I mentioned previously, some variations are known to have clinical implications, either from specific studies or from large-scale association studies called GWAS, which we will mention in a later unit. In such cases, there is a link to another database run by the NCBI called ClinVar, which details the evidence for the clinical consequences of the variant. dbSNP catalogs changes of a single or small number of nucleotides, while larger genomic variations, which span longer regions of DNA, are stored in a parallel database called dbVar. Another clinically oriented genomic database is OMIM, Online Mendelian Inheritance in Men, which was established by the pioneering work of Victor McCossick as early as 1966, and which mainly covers genes and phenotypes that are involved in Mendelian genetic disorders. More than 8,000 such disorders are curated in this database. how can we explore the wealth of information that is contained in these biological databases? One of the most useful ways to use this information is to employ a genomic browser, a graphical interface that starts with a DNA genomic sequence and enrich it with layers, known as tracks, of additional information. One such browser, and the most popular one, is the UCSC genomic browser from the University of California, Santa Cruz. The first layer shows the sequence of the genome. Here we concentrate on the human genome, but the browser can show information regarding dozens of organisms. The browser lets the user scan the sequence of each chromosome, zooming in or out to regions of interest. Below the DNA sequence, one can see the gene structure of the region, i.e., location of the exons, introns, and control regions. Then there is a track showing the protein sequence that can be translated from the underlying open reading frame. There are expression tracks that indicate the organs or tissues in which the gene is expressed. There are conservation tracks that show the sequence of homologous protein in other organisms. As we will discuss in a later unit, analysis of conservation patterns is a key step in determining whether a variation is likely to be deleterious. There are also tracks to show known SNPs and variants that were identified in the explored genomic regions, as well as tracks showing the connection to phenotypes and disease with link to the relevant literature. The selection of tracks appearing on the user screen can be tailored to match the need of each user. Genomic browsers, and the UCSC in particular, have become among the most useful tools that the bioinformatics community, in a collective effort, can offer to biological and medical research. UCSC has created several excellent publications and tutorials to facilitate the use of this tool, and we will walk you through some of the most useful features in the practice lesson. In the next unit, we will present next-generation sequencing, the technology that revolutionized genomic research, and discuss its application to medicine. So, see you next time.